Hi, this is Whitney, and I'm going to be extracting DNA from a banana today. We use a soft fruit for easy DNA extraction because since the fruit is very soft, we are more easily able to crush the cell walls when we smash the fruit and thus get the DNA, which is stored inside the nucleus of the plant cells. In order to extract the DNA from the banana, we have to isolate the DNA from all of the other parts of the plant cells, so the mitochondria, the lysosomes, the Golgi apparatus, the nuclear membranes, etc. I'm going to go over a list of all the materials that I used in this experiment. So first, I used measuring cups and measuring spoons, just like this one. I also used a funnel, which I fashioned out of a soda bottle, and some cheesecloth, which I attached with a rubber band, and also a glass, which I'm going to put it in. So you can see that here. And then I made some extraction liquid, which you can see right here. And this is what I'm going to use to get the DNA out of the banana cells. So this liquid contains one half of a teaspoon of salt, which is NaCl, sodium chloride, so just table salt, a third cup of water, which we all know is H2O, and about a tablespoon of dish soap, which is why it's blue. I'm using a banana. I live on Guam and I couldn't find any strawberries, and a Ziplock bag. And what I've done is I cut up another banana into tiny pieces, and I have already crushed it up using my fingers just like this. I smashed it up for 3 minutes, just so you didn't have to watch me smash a banana for 3 minutes because it wasn't very interesting. And again, that is how we're able to break down the cell walls and get to the DNA, is by manually smashing this banana. So this is what about one smashed banana looks like in a Ziploc bag, and I squeezed all the air out of it. And then a test tube. I couldn't actually find a test tube, so I have this shot glass here, which I think will work just fine, and a bamboo skewer, which is how we're going to take the DNA out once we've precipitated it, and also rubbing alcohol, so isopropyl alcohol, which is C3H8O, which we know is 3 atoms of carbon, 8 atoms of hydrogen, and 1 single atom of oxygen. And I've had this in the freezer overnight, so it is pretty cold. And that's what we're going to use to precipitate our DNA and turn it into a solid. So the very first thing that we're going to do is we're going to pour our mixture into it. But first what we have to do, now that we have our smashed banana in here, it smells really good, like smashed banana, we are going to take 3 tablespoons of this extraction fluid and add it to this mixture. So I'm adding 1 tablespoon. So this is just going in with the bananas. Two tablespoons. And 3 tablespoons. So we now have a mixture of smashed banana and extraction fluid. And I'm going to really carefully, trying not to spill this all over the place, seal up this bag and take out the extra air while I do it. So there we go. And now I'm going to basically mix this by smashing it for about a minute. And while I do that, I want to tell you a little bit about why we use the stuff that's in this extraction fluid. So in here, we've got water, and then we've also got salt and dishwashing soap. So the soap is able to kind of chemically break the cell membranes, so whatever's left of the cell membranes, by dissolving the proteins and lipids that form the membrane. So we've already broken the cell wall mechanically, and now we are chemically breaking the membranes with the soap. So it's dissolving the proteins and the lipids, and once we've kind of broken the nuclear membrane, then we're able to start extracting the DNA, which is stored in the nucleus. And what the salt molecules do is they can negate the negative charge of the DNA strands, and they make the strands clump together, which is how we're able to see them. All right, so this seems like it's pretty well mixed. And now what I'm going to do is I'm going to take all of this and I'm going to very carefully, not spill it everywhere, and pour it into this funnel right here. So because I have the cheesecloth in here, what's happening is all of the liquid is dripping down through the cheesecloth, but the solids, so the other parts of the cell that we don't need when we're just trying to extract the DNA, they are going to stay up in the top of the cheesecloth. So we're just getting the liquid. And we'll just give that a couple more minutes to filter through. So you can see here. And we've already got quite a bit of liquid down here. So what we're going to do next is I'm going to, I'll wait until this is pretty much filtered out and it's stopped dripping liquid down into the bottom, and I will pour it into this shot glass / test tube. And then what we're going to do is we are going to pour very slowly, take some of this really cold isopropyl rubbing alcohol and pour it really slowly down the side. And what that's going to do is it's going to form a layer of alcohol on top of the banana mixture, and in between those two layers is where we're going to find the DNA, which is what contains the banana genes. The reason we use the isopropyl rubbing alcohol is because it will dissolve everything in this mixture except for the DNA. So it will also extract the water out of the DNA molecules. It causes the molecules to collapse and then they'll precipitate into solids. And this will allow us to pull out the strands of DNA using this bamboo skewer. And the reason why we use the really cold isopropyl alcohol, so it's been in the freezer, is because that increases the amount of DNA that can be precipitated. So we've got a pretty good amount down here, and it's kind of stopped dripping. So I'm going to go ahead and take out this funnel, and we don't need this anymore. And now, here's my mixture. So I'm going to pour it into this container, so it's about 1/4 full. Let's see. That looks like a pretty good amount. All right. Oops. So here's what we've got. All right, so here I just stirred it up a little bit. This liquid contains our DNA, and now I'm going to very, very slowly pour this rubbing alcohol down the side. I'm doing this really, really slowly so I don't mix the two liquids. All right. So what we can see here is we very clearly got two layers, and we have some stringy things that are kind of starting to pull up and separate and come into the alcohol, and that is our DNA. So this is the DNA of the banana. I'm not sure if you can see here, but I've got these strings. They're a little small, and they're clumping on the skewer. So these are these white strings of this isolated DNA that came out and was precipitated by the cold rubbing alcohol. So DNA is the blueprint for life because it holds all of the genetic codes that describe how an organism will grow, live, and reproduce. DNA is essentially a recipe for making and growing an organism. This is because DNA controls protein synthesis in cells, and this is a really vital function since proteins are the basic unit for cell function and structure. Well, I hope that you enjoyed this experiment. Thanks for watching.